And welcome back. Now we're going to talk about the cell types of the nervous system, and there are two major types of cells. There are neurons. These are the structural units that we typically think about when we talk about the nervous system. They are highly excitable, and they conduct information from one cell to another cell. And then we have glial cells or neuroglial cells. These are not excitable cells, but they are very important. They outnumber neurons, and they protect and help the neurons do their jobs. Here's a neuron. This is a common structure for a neuron. So we have dendrites up here that take in information and send it to the cell body. Here's the cell body, where we process information. Cell body is where the nucleus is going to be found, as well as all the other organelles. And then we have this axon. The axon is the region where information leaves the cell body and moves to neighboring cells. This region right here would be called the axon hillock. The hillock is where the axon emerges from the cell body. And it's also the region where the majority of action potentials will start. Most of our neurons have a myelin sheath. Myelin is a fatty tissue that wraps around the axon and basically insulates and increases the rapidity of an action potential, so it makes it much more fast. In between the myelin sheaths, there are tiny little nodes called nodes of Ranvier. This is where action potentials actually jump from one area to another area, which we'll talk about when we're talking about the myelination a little bit later. And then at the end, we have the axon terminals, which have these tiny little synaptic bulbs that contain neurotransmitters. Neurons can be classified structurally by the number of processes that come off the cell body. The most common type of neuron is the multipolar neuron. Most of the CNS neurons are multipolar, and a multipolar neuron has multiple poles off the cell body. So here's 1, 2, 3, 4, 5, 6 dendrites and 1 axon. A bipolar neuron, which we see in the neurons of olfaction as well as neurons of sight, are going to have 2 poles off the cell body. We have one that is the dendrites and one that is for the axon. So here we have the dendrite and here we have the axon. Unipolar neurons are found in the PNS. They often are the touch receptors that we have that take in sensory information from our skin. So here we have dendrites pulling in information. They go up this one single pole. The information is processed and moves down and out through the axon over here. And so let's look at how information moves through the nervous system. So functionally, we have sensory neurons that take in information, and in this case, we have, we're looking at the temperature of water. So we have sensory nerves taking in information, will integrate the information in the brain, and then bring about some change through the efferent system. So sensory neurons take in sensory information from receptors. They pull that information in and bring it to the CNS. Most of these are those unipolar neurons, but we do have bipolar neurons in our eyes, in our nasal cavity. And so here we have this neuron receptor pulling in information. It's going to go to the cell body, be processed, and move to through the axon to the CNS. The CNS is where integration occurs. So CNS can be in the spinal cord or in the brain. It receives information and determines what has to happen. So how do we respond to the information that was given? These are almost always multipolar neurons, and this is where the majority of our neurons are found. And then we have the motor division, which sends information out. So once we have decided what has to happen, then a motor neuron is going to send information through the efferent or motor division to the muscles or glands, in this case muscles, to tell us to change the temperature so we can then have the temperature at whatever temperature we want the water to be at. Neuroglial cells then are the more common cells of the nervous system, though they are not nearly as talked about as your neurons. So they do outnumber neurons. They are not excitatory, so they don't send signals from one cell to another like your neurons do, but they are able to undergo mitosis, so when they get damaged, they can be fixed. Their function is for support and protection of the neurons. They help in binding neurons together. They help in producing some of the pathways and making sure that the neurons are able to fire properly. So here's a picture of the 4 glial cells that are found in the central nervous system. We have astrocytes, we have microglial cells, we have ependymal cells, and we have oligodendrocytes out here. So there's one. So astrocytes are probably the most abundant of all of our neuroglial cells of the CNS. They surround neurons. They form a barrier between the blood capillaries and the neurons. So they form what we call the blood-brain barrier. And so they can help in controlling the chemical environment of the brain, making sure toxins don't get in, stuff like that. Ependymal cells, they help in forming the choroid plexus, and the choroid plexus functions in producing cerebrospinal fluid. So ependymal cells help in producing and circulating cerebrospinal fluid, which is needed to maintain a healthy brain and spinal cord. Microglial cells are the macrophages of the nervous system, so they wander around and degrade any potential damaged material and damaging agents like pathogens. Keep us healthy. And then the oligodendrocytes are your myelin sheath producers of the CNS. So they wrap around multiple neurons. This helps to bind the neurons together as well, and help increase the speed of an action potential. In the PNS, then, we have satellite cells and Schwann cells. Satellite cells are your cells that wrap around the cell body, and they help to regulate the chemical environment and to protect the cell body. And then we have the Schwann cells, also known as neurolemocytes, that form myelin sheaths of the PNS to help in propagation of action potentials. So myelination of the axons occurs by either the oligodendrocytes or the Schwann cells. And when they do, they wrap around, forming a fatty barrier between the neuron and the and the environment around that neuron axon. What this does is it allows for information to be processed faster because we don't have to worry about this area that's been wrapped with a neuron or with a myelin sheath. We can move from here to the next region of the axon. Oligodendrocytes will wrap multiple areas of more than one axon, whereas Schwann cells, they produce one small myelin sheath per, so you have to have multiple Schwann cells to myelinate an entire axon. I'm going to stop here for a second. Hold on a second. So why is myelin important, you might wonder? When you think about an action potential moving along an axon, so these neurons are relatively long. These axons can be extremely long. They can run the length of your leg. And so when an action potential is moving along that axon, it's going to have to open sodium-gated channels all along the axon. So we don't just have depolarization of the entire length of the axon at one time. It goes in stages. So you have depolarization here, and then you have depolarization here, and then you have depolarization here, and then here. And as depolarization is occurring down, you have repolarization occurring. So everything is happening. You have depolarization, then depolarization, and repolarization occurs here. It's kind of like the wave. Think of it that way, okay, when you do the wave. If you had to move and open every single sodium-gated channel on this axon, it's going to take a long time. So think about walking from one side of a block to the opposite end of the block, how long it would take. If we wrap myelin around some of that axon, we decrease the area for sodium channels to have to open. So now, instead of moving through the entire length, we only have to open up sodium-gated channels here, 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 and there to get to the end. So this would kind of be like running down the block, so much faster. Or if you're handing a ball across from one student to the next student all the way down to the end versus tossing the ball from one table to the next table to the next table to the end. It's going to be a lot faster. So, you can think of this as the analogy of the tortoise and the hare. The hare moves much faster than the tortoise. I do know that in the actual story, the hare keeps taking naps, but we're not looking at that. We're just looking at, in general, the speed. So this movement from one area to the next node is called saltatory propagation. So we're jumping from one area to the next versus crawling along. Okay. So I'm going to stop here, and the next video we'll talk about multiple sclerosis.